Welcome back, everyone. This is part 2 of our discussion of sections 4.1 and 4.2 of the introductory microbiology text. We're going to pick up where we left off in part 1 with additional structures that we find within the eukaryotic cell. And the next structure we're going to talk about is the endoplasmic reticulum, sometimes shortened as ER. It is a series of tubes and tunnels sort of within the cell, very closely connected to the nucleus and nuclear membrane that assists in the transport and storage of proteins and lipids. So there are two forms of endoplasmic reticulum. We have the rough and the smooth. And the rough ER is primarily for storage and transport of proteins. And it is called rough because externally it is covered in ribosomes. And those ribosomes make it appear very rough or bumpy. And those ribosomes attached to the membrane of the rough ER are constantly interpreting the message in messenger RNA and synthesizing proteins that are then transported within this membrane network of the rough ER. The other form of endoplasmic reticulum is called the smooth ER. And it is smooth because it does not have ribosomes attached, and it is primarily a location for storage of lipids. It is not involved in protein synthesis or storage because those ribosomes are missing. The Golgi apparatus, or the Golgi body, is a series of stacked flattened membranes that are then involved in the modification of those proteins that are synthesized in the endoplasmic reticulum. The Golgi is going to receive these transitional vesicles that are transporting synthesized proteins from the ER to the Golgi. And within the membrane of the Golgi body, then we're going to modify those proteins with polysaccharides, sometimes lipids, and those proteins will be packaged within a condensing vesicle, and that vesicle will pinch off the end of the membrane, or that cisternae, and then those condensing vesicles will then travel through the cytoplasm and connect with another organelle or the plasma membrane to either be transported outside of the cell or used within the cell. So this protein assembly involves the nucleus, the ER, and the Golgi. And the way that protein is synthesized, and we'll get more into the details of this process later in the course, but the general idea is that the gene, or the message that's contained within the DNA, is transcribed into another molecule called RNA. And that RNA is messenger RNA. That messenger RNA carries the message from the nucleus through the nuclear pore and into the cytoplasm, where it is going to meet a ribosome. That ribosome, with the help of ribosomal RNA and transfer RNA, is going to interpret that or translate the message within the messenger RNA and synthesize proteins that are going to be stored and travel through the ER to the Golgi apparatus, where they will be chemically modified, packaged, and then transported to where they are needed within the cell. Lysosomes are another organelle that we find within eukaryotic cells. These organelles contain digestive enzymes, and these enzymes are involved in the breakdown of food molecules. They also protect the cell against structures that are not self, so any invaders, microorganisms, viruses, and they also participate in breaking down and removing organelles and other damaged tissue within the cell. Vacuoles are membrane-bound sacs within the cell that can contain fluids. They can also contain solid particles that need to be either digested, excreted out of the cell, or stored. Vacuoles are formed in phagocytic cells like our macrophages in response to food and other substances that have been engulfed. And then generally a vacuole and a lysosome will merge. So the digestive enzymes within the lysosome can then combine with the contents of the vacuole and begin to break it down. And that's what this diagram shows, is a vacuole that has a material that needs to be digested, and then that merger between the vacuole and the lysosome breaks down whatever is in the vacuole, and then it can then be digested and used by the cell. So one of the key structures in eukaryotic cells is the mitochondria. And the mitochondria's primary function is the generation of energy. Mitochondria are unique in that it is composed of two membranes. You have this external outer membrane, and then you have a very folded inner membrane. And those folds are called the cristae, and this is where a lot of the chemical reactions for ATP synthesis occur. Mitochondria extract chemical energy, ATP, from glucose. Mitochondria are also unique in that they contain their own ribosomes. They are 70S ribosomes, so they are very similar to bacteria ribosomes. They also contain their own DNA, and this DNA is circular, like bacterial DNA, not linear. And they are able to divide independently of the eukaryotic cell itself. So that information there, that evidence helps to support that endosymbiotic theory that mitochondria were once bacteria that were engulfed by eukaryotic cells. Another organelle found in eukaryotic cells is chloroplast, and they are only found in photosynthetic cells, so algae, plant cells. They are capable of converting the energy in sunlight into glucose, food, and they produce oxygen as a waste product or a byproduct of that process. Again, chloroplasts are also composed of a double membrane. So they have an outer membrane and an inner membrane. And then these stacks, the granum stacks of thylakoids, this is where a number of the reactions necessary for photosynthesis take place. They are somewhat similar to mitochondria, but larger, and the pigments they contain are what help to transform that light energy into food. So our next structure that we're going to encounter within eukaryotic cells are ribosomes. And remember, bacteria and archaea also have ribosomes, but they are different from those found in eukaryotic cells. We find ribosomes scattered freely throughout eukaryotic cytoplasm, and they are also found attached to the rough ER, and we also find them inside mitochondria as well as chloroplasts. Their composition is of two subunits, a large 60S subunit and then a small 40S subunit. And when you combine them, it forms a total 80S ribosome, which is different from those of prokaryotic, the bacteria and archaea cells, which are 70S, a 50S large subunit, and a 30S small subunit. And ribosomes are the primary organelle for protein synthesis. Other structures we're going to encounter are the cytoskeleton, and the cytoskeleton is responsible for providing structure, adjusting the shape of the cell, providing movement. It's also involved in anchoring organelles within the cytoplasm and moving organelles within the cytoplasm. There are three types of filaments that are found as components of the cytoskeleton. You have actin filaments, which are very long protein strands. We also have microtubules, which are long hollow tubes. These are the components of the flagella. And then we have structures called intermediate filaments that are rope-like. They are thinner than actin filaments and typically shorter. So that's where we're going to end part 2. Make sure you go through these questions and quiz yourself. Be certain that you go back and look at the objectives and make sure that you understand all of the concepts presented here. Hopefully this was helpful.